Hello, I'm talking about contrastive mixture of posteriors for counterfactual inference, data integration, and fairness. My name's Adam Foster. This is joint work with R.P. Vaser, Craig Glastonbury, Paudie Creed, Sam Abujada, and Aaron Sim. And we started this work when we were together at Benevolent AI. We're working with omics data. So to give a quick example, take a matrix in which rows represent cells or samples, columns represent genes, and the value tells you how highly expressed that gene was in that cell. We were particularly interested in transcriptomics and single-cell RNA-seq. And we're looking at representation learning for omics data. So that means for each cell or each row in the data set, we produce a latent representation. So one point in the latent space represents one cell. And this latent representation could be used to find hidden structure in the data, or as we'll see, it can be used to tackle certain important problems in computational biology that can't be solved directly in gene space. And one of those key challenges is data integration. So we have many data sets, and we'd like to combine them together to get more out of our data. But often these data sets have technical variation between them. They might have been prepared with different library protocols, for example. And that means just directly concatenating these data sets is not going to be helpful. And we get the same problem when we're combining tumors with their corresponding cell lines. And here we can see that illustrated on a latent space plot. So blue points are tumor samples and orange are cell lines. And we know that these data sets do contain comparable cells. Both contain prostate cancer cells, breast cancer cells, etc. But there's this unwanted global variation, which means that they don't overlap. They're not comparable on this latent space. So we want to subtract out this tumor cell line global variation and do data integration. The other problem we're looking at is counterfactual predictions of the effects of interventions. So we have a real cell here, and we've observed its transcriptome in our data set. But we would like to know, how would this real cell have looked? How would its transcriptome have looked if it had had some knockout applied? So that's a counterfactual prediction problem. And we can use similar technology, we'll see, to answer this problem. And finally, we're looking at learning fair representations. This takes us a bit away from omics data. So this fair representation is a representation of each row of the data set, which now might represent an individual person, such that these representations could not be used to recover a sensitive attribute about those people. And the conditional variational autoencoder framework is the computational setting that we're going to use. That's the representation learning sort of basis that we'll use in this project. So a CVAE, it takes as input the transcriptome X and some condition label C. So that is how we make connection with these key problems. So for batch correction, that would be an experimental batch label. For counterfactual inference, that would tell you the condition, the intervention under which the sample X was collected. In fair representations, that would be the protected attribute. These are encoded to produce this latent representation Z. And we also have a decoder network that takes the condition and Z, the latent representation, and it reconstructs the original transcriptome. And there's one more condition or requirement that we haven't mentioned, and that is that Z and C should be modeled as independent variables. And what does this look like in the latent space? Well, going back to our tumor cell line example, if we color two different conditions in different colors, then we should see, if Z and C are independent, we should see perfect alignment in latent space between these two colors. And we can already see how this is useful for data integration. We've essentially done the data integration task in the latent space by enforcing this requirement Z independent of C. But the problem is if we just train a CVAE using the standard elbow objective, the model may not even use the condition label C. And on this tumor cell line dataset, we can see we do not get good alignment when we just train the network using the standard objective with no additional tricks. And that's where our project comes in. Contrastive mixture of posteriors is a misalignment penalty, and we will use that to train a CVAE such that Z independent of C does hold. And actually this plot with good alignment, this is an experimental outcome using comp on the tumor cell line data. So the way we achieved this, we started by rewriting the requirement in terms of the KL divergence. So we want the KL divergence of the blue distribution, so that's cells in condition C, to be equal, so to have 0 KL divergence with the distribution of orange cells, which is cells under any other condition. And this inspires the form of our comp penalty. So let's break this down. This is a bit of an unfriendly formula. So we start with representations of one training batch. So these are random cells sampled from the data. And we form their representations. And we have two terms in the penalty. The first is a repulsive term, which says that points with the same condition should be pushed further apart. And that's defined in terms of the encoded distribution Q. And secondly, an attractive term, which says that points with different conditions should be pulled closer together. And again, this is in terms of a mixture of the encoded distribution. And the combination of these should encourage different conditions to align nicely in the latent space. We proved an important theorem about this penalty. The expectation of the comp misalignment penalty is an upper bound on this weighted sum of KL divergences. So this links our penalty back to the original KL idea, and it shows that it's an upper bound. So if we minimize this upper bound, we should be minimizing the underlying divergences. So our overall idea is to just train a CVAE model, same networks, but the objective function is no longer just an elbow. We subtract a scaled version of the comp penalty. So let's look at applying a trained comp model to some of the key challenges we described at the beginning. So for data integration, this is quite clear. We encode X and C to Z, and the requirement Z independent of C is equivalent to saying that different batches should be aligned, so we can use Z as a data integrated representation. For counterfactual inference, it's slightly more involved. So we take the observed transcriptome and the observed intervention that was used to prepare this sample. So here we encode with drug equals yes. Then we obtain our latent representation. Now, to make a counterfactual prediction, what would this cell have looked like if it had not had the drug? We swap the label to drug equals no, and then we decode according to this new label drug equals no. And instead of a reconstructed transcript, what we get is a counterfactual prediction of the transcript in the drug equals no case. Now, to get counterfactual predictions, we actually need to think a bit more about the underlying theory. In particular, is this requirement Z independent of C sufficient to identify counterfactuals, assuming the model is correct? Unfortunately, the answer is no. And we can see that with a simple counterexample. So here's a latent space in which Z and C are not independent. But if we apply a simple right translation to these orange points, then we'll have good alignment and overlap, and Z is independent of C. But we could also achieve that alignment in another way by doing a 180-degree rotation of these orange points. And again, we end up with Z and C independent with good alignment. So What's the problem? The problem is that we cannot tell which of these alignments is the correct one. And if we attach a letter label to these 4 clusters, we can see that with translation, A and C are aligned, but with rotation, A and D are aligned, and there's no way to tell which of these models is the right one. But there is some good news in terms of a theorem we proved in this paper. That says, if we have a linear decoder and we assume that there is no exact linear symmetry of the true latent distribution, then counterfactuals are identifiable. Again, let's look at this in a very simple example. So here we have almost the same latent space, except the symmetry between A and B is broken. And now, because there's no symmetry, there's one unique way to align them. Here it's by doing a translation, and that means that we can identify the right alignment, and we can identify counterfactuals. In cell biology, exact symmetries are rarely seen in practice, so I think the take-home message is counterfactuals are likely to be identifiable in most practical cases. Let's conclude with some results. So for data integration, this is the tumor cell line data set that I've been showing throughout the talk. On the left, we have fully uncorrected. The middle is an existing method called CellLiner. And on the right, we have our method, which achieves, visually the best overlap, the best alignment. And these metrics confirm in the left 2 columns the best overall alignment. And in the right 2 columns, we introduce these cell type labels. They're not used during training. And we show that comp is best at matching the correct cell types in the 2 conditions together. For example, here's a prostate cancer tumor cluster. And for comp, we can see some prostate cancer cell lines correctly aligned, and a few other cell lines have been misaligned and placed in this cluster. But with the previous state of the art, we can see a much larger number of misaligned cell lines. So we, also qualitatively get the results we would expect. Here is an example of single-cell RNA sequence data integration comparing against Seurat and Harmony, 2 of the standard methods. And visually, in the bottom row, we can see all methods get good overlap, although comp maybe looks slightly better aligned. And the metrics confirm that comp is the best, both in terms of global alignment and in terms of aligning cell types correctly together. Now we switch to counterfactual inference, and the problem here is based on blood cells. So we trained comp on a blood cell data set, and then we used comp to predict the expression levels of certain well-known differentially expressed genes when a drug is applied. And here we've tried to predict the effect of IFN beta stimulation on control cells. We looked at 2 cell types, DC and NK, and here a perfect prediction would be 0, so neither sort of over or under predicted. And we can see comp clearly gets the prediction error closest to 0 of any of these methods. And finally, we looked at fair representation learning on the adult income data set. Here, the protected attribute is gender, and we can see visually comp gives us the best mixing between the 2 genders in latent space. And if we look at this income accuracy column, this quantifies the amount of useful information that's still in the data or still in the representation. And comp is comparable with all these other methods. And in terms of gender accuracy, so that's trying to work backwards from the representation to gender, which should be difficult, this value is essentially equivalent to random guessing. So we've achieved our aim. So that just leaves me to say a big thank you for listening. Thank you to RP, Craig, Pordy, Sam, and Aaron. Our code is available, and please check out our paper for a lot more detail.